Thank you, Tara. Before starting, the two presenters are myself and Dodwa, but Michael is here, and I'm grateful to have Michael here because uh, him and Dodwa were the two main investigators in this project, and they did the field work and collected the data. And he's very well positioned to answer some of the most difficult questions you will have. And one person who's not here is Ricky Kong. Ricky was part of this project from day one and worked mostly on the greenhouse part of this. And you will be seeing Ricky next month for the last webinar. Next, Dodwa. And before starting, I will take the opportunity here to thank arckp for giving us the opportunity to work on this project, and thank them just because they help us to create a relationship between Portage College and NATE, a relationship that we didn't have before. And Michael Schultz is here representing Portage College, and Dodo and myself, we represent NATE. We created a relationship that can last very long going forward. And the funding that we received from arckp was further leveraged by NSERC Applied Research Grant that help us expanding the scope of this project and also the time. And I also appreciate all the information we receive from many forest companies, West Fraser in particular, as they open their long time lichen transplantation site to us for another study, and also open lots of cat blocks around Wycott and Fox Creek to set up our trials. Next, Dua. So this presentation has two parts. The introduction part is what I will be talking about, and Dua will take on starting on the lichen transplantation. So the introduction part will be only about the background of this project, and all the data, methodology, and objective of the project will be on the second part that Dua will be presenting. Next. So just to give you an idea, the study we will be presenting on today is part of a large project, titled Feasibility of Lichen Transplantation. And we started this almost 4 years ago. And this project, we partition it into teams. And the very first part, something we did in the early months of the project, was to do a literature review, a general literature review that helped us to understand the current state of research on lichen transplantations. We wanted to know what other people are doing or have done in Alberta or in Canada or elsewhere. And we found some study actually in Europe. And these studies help us to design or to redesign our greenhouse trial. And that greenhouse trial was basically to test different lichen fragments and on different substrates. And the result of that greenhouse trial will be the subject of our next webinar next month. Again, it's not scheduled yet, but it will be next month. It will be presented by Ricky Kong. And so after setting up the greenhouse trial, we started a field trial that was some sort of copy and paste trial of the greenhouse. That means the same thing we're testing in the greenhouse is what we set up in the field. And as we were doing that, we had go to the next slide, Dua. As we were trying to set up trial in the field, we also went and we made some sort of inventory of existing lichen transplantation trials in Alberta and BC. If you remember, for the people who were here at the last two webinars, we presented on a trial in Hinton. It was a long-term trial, 21 years old, I guess, and two other trials in BC that were set up by Chucho Environmental and the government of BC. So these three trials also help us to understand the challenges that other people have faced and the methodology that they are using for lichen transplantation. And that led to the study we are presenting the result today, which was, as I mentioned, to test this time at the harvest block level, but at the plot scale, not at the field site scale, to test the effect of lichen fragment size, the substrate, on the lichen survival and health. Next slide, Eduardo. And why do we care? Many studies have shown that caribou population is in decline in Alberta and somewhere else. And this is mainly due to the alteration of their habitats. And forest activities is part of those activities that alterate the habitat of woodland caribous. And the second thing was that caribou feed a lot on lichen, especially during the winter months. And some studies have demonstrated that up to 60 to 80% of the diet of lichen during the winter comes from lichens. And the other importance of the lichens is that it's a good indicator of the pollutions. It's helped us to understand also the environmental health in the forest setting. Next, Zada. Next slide. Yeah, and some knowledge gap, I think I mentioned this, some knowledge gap that we have when it comes to lichen transplantation sites is, what is the fragment size that the optimum fragment size that could be used to really conduct lichen transplantation in the forest setting. And what are the substrate type that we need? We know where the lichens grow in the forest, but we don't really know after forest activities if these substrates are still the right substrate to do lichen transplantation. And some studies have shown that lichen doesn't actually spread very fast. So there is a limitation in the dispersal distance of lichen. So all these are challenges that our research is trying to help understand what we can do to actually transplant lichen at harvest block level. And for our particular project, I will now pass on to Duadua to give you the objective, present the results, and we can discuss them.
Yeah. Thank you, Jean-Marie. So the research objectives of our study are to assess the variability of terrestrial lichen fragments of different size, to establish growth on different substrates, identify the most suitable substrates for promoting the re-establishment of terrestrial lichens in area disturbed by forestry management, and examine the relationship between the success of lichen transplanting and the cover of non-target lichens, mosses, forbs, plants, and graminoids. This picture here shows our 1 meter by 1 meter square research plot, siting up in the harvest block. So first, we collect lichen samples and substrate. Lichen samples were collected from bog, swamp, and pine forest around Peace River in 2021 and Fox Creek in 2022. So when we collect lichen samples, we consider not damaging the original growth habitat. As this picture showed, we lay out the 1 meter by 1 meter quadra in high lichen density sites, selected to represent the diversity of lichen species present in each area. And as the first picture shows that, we also collect the ecological site informations in that area. I did not include the data here, but it is available for future use. So all lichen samples were collected by hand and placed in paper bag and transferred to our lab. And in the lab, we cleaned them and allowed them to air dry completely. And then they were stored in paper bag at room temperature in the dark. We also collect the substrates of pine needles, woody debris, and mosses near the area that we set up our research plots. So this map shows the trails the trails were set up in the 6 conifer forest harvest block between Fox Creek and White Court in June 2022. These sites were chosen to represent a diversity of forestry harvest block treatment. So B and G has been modified as a replot. D and one side of E was modified by slashing. One side of E has been modified by mounding. Sides C and F were untreated. However, in the spring of 2023, wildfire burned through sites C and D. As a result, the number of sites in 2023 and 2024 were reduced from 6 to 4. So in each site, we have 2 transects, and in each transect, we include 12 1 meter by 1 meter square plots, as the picture showed here. And each plot include treatment combines of the 4 substrate, soil, moss, pine needles, and woody debris, treated with one of the 3 fragment size, small, medium, and large. The soil treatment include only the bare base layer of soil, with the later layer manually removed. In some cases, they were mineral soil, and in others, they were peat, due to the type of the soil present on that site. For the moss treatment, a layer of leaf moss about 5 centimeter deep was transferred to the plots. The pine needle substrate consists of a layer about 2 centimeter deep of logical pine needles spread evenly in the plot. The woody debris treatment consists of down woody materials about 5 to 15 centimeter in diameter, placed tightly within the plot. So the dried lichens was fragmented by hand into categories of small, which is 0 to 2 centimeter, medium 2 to 4 centimeter, and large 4 to 6 centimeter. Then about 40 grams of air dry stellaris fragments were dispersed evenly within each plot by hand. We collected lichen samples from field in the summer in 3 years from 2022 to 2024. So as this picture showed, we throw the 10 centimeter by 10 centimeter quadra into the plot and randomly select the fragment that closed to the center of the quadra that was within the target size for this plot. We place them into the envelope and label them for transportation into the lab. So a maximum of 5 samples were collected in each plot. In 2024, we also assessed a lichen cover using the same size of the quadra and the percentage cover of all plant species within each plot. To obtain an indication of lichen fragment survival and health, all collected fragments were measured for chlorophyll fluorescence. So we sent the sample to the University of Northern British Columbia Coxon Research Group. They rehydrate all sali by spraying them with the eye water. The sample were kept in a container and saran wrap sitting on a damp paper towel in the light at moderate illumination of 200 micromole per square per second, which is a common light intensity used for growing plants and microalgae, and temperature of 15 degrees for 24 minutes. So they use a device like this picture showed, and the chlorophyll fluorescence parameter FVFM was recorded within pulse-modulated chlorophyll fluorescence unit within a 6 millimeter measure disk after a 5 minutes period of dark adaptation. So based on the description in public paper, the lichen samples with a value over 0.7 treated as a healthy sample, and the samples with a value less than 0.7 were under stress. If the sample with a value 0, it means they are died. Lichen species was identified when we collected them in the field using hand lens. And then after chlorophyll fluorescence analysis, we confirmed the identification in the lab using the microscope and chemical spot testing. So we focus on 6 species in our sample, which is Cladonia mitis, C. rangiferina, C. stellaris, C. uncialis, C. stygia. And one that I did not show here is Stereocaulon tomentosum. Now we know to the results of this study. The first one I want to talk is lichen survival. So the survival of lichen fragment collected from each plot was calculated as the number of the lichen fragment having FVFM value over 0 divided by the total number of samples with available FVFM value. So differences in the survival of lichen fragments were analyzed using GRM-TMB function and auto-beta distribution with year, substrate, size, and their interaction as fixed factors, and plot nesting into transects and transect nesting into sites as a random factor. So the result shows that the effect of size and years was significant on lichen survival. However, pairwise comparison did not indicate a significant difference between years, as the first figure showed here. The survival rate of lichen fragments varied between 65.4% and 69.6%. The large lichen fragments had a significant higher survival rate than the small one, and the medium had no significant difference on survival rate compared to the small one and the larger one. We also analyzed lichen survivals for each species. Survival rate for each species different March. Use pooled data from 2023 and 2024. Survival rate of C. stellaris and C. rangiferina were moderate at 80.4% and 73% and the survival rate of S. tomentosum, C. uncialis, and C. mitis were between 92% and 98.3%. However, the C. stygia had a low survival at 55.2%. Unfortunately, we do not have enough replicates in some treatment, so we could not do the statistic analysis on survival rate for each species by substrate and fragment size. Now we move to the results of lichen health. The health of survival lichen fragments indicated by the mean FVFM values, which was the mean of all FVFM value over 0 within the plots. So we use the same function and distribution in R that we used for lichen survival. The results show that substrate, year, and the interaction between size and year had a significant effect on the mean FVFM value of alive lichen fragments. So this figure shows that lichen fragment showed exhibited significantly higher mean VFM value on soil and pine needle substrate than the moss. But lichen fragment on all substrate were within the healthy range. There was no significant difference on FVFM value across among the 3 fragment size in 2023 and 2024. The mean FVFM value of all lichen fragments measured in 2023 was significantly higher than those recorded in 2022 and 2024, except for the small-sized fragment in 2022. We also analyzed the lichen health for each species, but sometimes we don't have enough replicate for some treatment of some species. I labeled it as NA, it means not available for this treatment in this species. The effect of the substrate fragment size and their interaction was not significant for the mean FVFM value of a live lichen fragment of C. mitis and C. rangiferina in both years. So I have no figures for these two. For C. uncialis, substrate had a significant effect on the mean FVFM value of the left lichen fragments collected in 2024. So as this figure showed, fragment on wood had significantly higher mean FVFM value than those on moss, but no significant difference from those on pine and soil. For stellaris, the effect of substrate size and interaction between substrate and size was significant on the mean FVFM value of a left lichen in 2024, but only the interaction had a significant effect on the value in 2023. And the peer-wise comparison did not show the significant difference, so I did not have figure for this interaction. But the data analysis for 2024 showed that small fragment of, say, stellaris on moss had significantly lower FVFM value than all, but not for the medium size on moss, small and larger size on wood. And you can see that there is no significant difference on FVFM value for all the treatment combined, except for the small fragment on moss. For stygia, substrate and the interaction between substrate and the size had a significant effect on mean FVFM of a left lichen fragment collected in 2023. But similar, the pairwise comparison did not show a significant difference across substrate or the interaction. So no figure for this one. For S. tomentosum, the effect of the size was significant on the mean FVFM value of a left lichen fragment collected in both 2023 and 2024. Peer-wise indicated the significant in 2023, but not 2024. In this picture, we can show that larger fragment of S. tomentosum has significantly higher mean FVFM value than small ones in 2023. So in 2024, we assessed the cover of all lichen species, all plant species in each plot, categorizing them into woody plants, forbs, gramineous, mosses, lichens, and we can see the result that the effect of the forbs and mosses was significant on the FVFM value, but not on lichen survival. And these two figures show the negative relationship between lichen health and the moss or forb covers, as the FVFM value you can see the FVFM value decreased as the cover of moss or forbs increased. This is maybe due to the high moisture caused by moss or forbs, and also the competition from them in the same plot. In 2024, we also collected the data of lichen cover. The results show no significant difference between the coverage on different substrate, but a significant difference between the fragment size. So the medium fragment size had a higher lichen cover, followed by the larger size, and the small size have the small lichen cover, but we did not collect the initial data for lichen cover in 2022. So it's hard to tell what's the condition at the beginning, and we could not analyze the effect of the fragment size and substrate on lichen cover changes from the beginning to 2024. So we will continue collect this data in the future. Now we move to the conclusion. Based on the results from our study, larger fragments preferable to small ones in lichen survival, while there were no differences in lichen survival between the different substrate. Living lichen fragments of different size were healthy on 4 substrates in the field, although surviving lichens on the moss substrate had significantly lower FVFM value than those on soil or pine needles. Living lichen FVFM value were negatively correlated with the cover of moss and fall. We also have some conclusion based on different lichen species in our study. So neither C. mitis nor C. rangiferina showed any significant effect of the fragment size or substrate. C. uncialis had significantly higher FVFM value on wood than on moss. C. stellaris showed a significant interaction effect of substrate and fragment size on FVFM values. The health of S. tomentosum was better than with larger fragments. We also have some suggestion for practice based on the effect of fragment size and substrate for the lichen transplanting. So the medium lichen fragments, which is 2 to 4 centimeter, had no significant difference from larger ones for survival and health, and could be used to provide more lichen covers. Substrate may not be a limiting factor for lichen transplanting on a larger scale. In terms of the lichen species we focus on in our study, C. mitis and C. rangiferina have the potential to be used for lichen transplanting on a larger scale because the effect of fragment size and substrate was not significant on their survival and health. The uncommon C. stygia had low survival and may not be a good candidate for lichen transplantation. As tomentosum had a high survival rate, and it is the only one species in our study that has a nitrogen-fixing cyanobacteria photobint. So it could be used in the area that needs nitrogen fixing. So this is all the summary from our study. Thank you for your attention.